now we are raising $20 million to support our clinical trials in the US. We have built a portfolio in oncology by identifying the most practical and promising asset for the new drug development. Starting with the radiosensitizer VS101, we have developing a radiosensitizer complex chemosensitizer. And there are migrastatic is a small molecule to inhibit the metastasis of cancer cells, and statutory inhibitor is under the development with the government grant. Today, I'm keep focusing on the radiosensitizer VS101. Firstly, I'd like to introduce why we develop a radiosensitizer. Conventional radiotherapy has been used for treating cancers by irradiating high-energy beams such as X-rays or gamma rays to the external body parts. It is efficient in killing tumor cells, but it has a problem. It impacts on the normal cells. So new technology have been adopted to advance the radiotherapy. They are different in irradiation types, but have something in common. They are targeting the tumor cells precisely. One of them is the radiopharmaceutical therapy. It is considered a groundbreaking technology in the field of in oncology. However, many key players are struggling with the challenges such as the limited efficacy and balancing safety and efficacy depending on the radioisotopes. So many people believe the combination of therapy is inevitable. To overcome these challenges, we believe that its full potential of radiotherapy should be awakened. We found solutions in the radiosensitizer. VS101 as a promising candidate for the radiosensitizer, accompanied with the benefits for patients. We aim to redefine the radiotherapy with the VS101. In 3 points, we believe VS101 will do it. Firstly, we confirmed its efficacy for various solid tumors in preclinical studies. Now we secured the first human data for head and neck cancer. And while that we have progressed in the mechanism of action studies, we confirmed VS101 has multiple mechanisms of actions while secured a clue to overcome the radio resistances. As we're proposing a drug, there is fewer concerns over the toxicity. We use the API has been used for over 20 years for human beings, and also we tested only 10% of doses for approved human bodies. This is how radiotherapy kills the tumor cells. Radiotherapy is known to induce the damages to the cancer cells in 2 ways. It directly breaks the DNA of tumor cells, while indirectly impacts on it through the RS generation. Adding to the VS101, we can expect the RS generation enhanced while also inhibiting the IAP, inhibitor of apoptosis proteins. Here, I'd like to elaborate on the IAP inhibition. IAP is a group of proteins that protect cancer cells from being destroyed by the anti-cancer treatment. We confirmed VS101 inhibit the various subtypes of IAPs. Here, important thing is the inhibition of survivin regarding the radioresistances. Survivin is considered as a predictive factor of treatment response of radiotherapy. High expression of survivin means that more likely to the radioresistances. This is our first IND-enabling data in the head and neck cancers. You can see here that adding the VS101 to RT, we can expect more increased tumor growth inhibition and also prolong the survival rate. The important thing is that VS101 is in fact effective only in combination with the radiotherapy. We got an IND approval from the US FDA for phase 2 using this data. I'm proud to present the first human data of VS101 for head and neck cancer. This is a part of the dry run analysis of phase 1 for head and neck cancer, which evaluates to the safety and tolerability of VS101. There are two cohorts, cohort 1 receiving 2 mg of VS101 plus RT, and cohort 2 receiving the 5 mg of plus RT. And we confirmed that there is no dose-limiting toxicity, and most of the adverse events are not related to the treatment. Moreover, there's no negative impact on the patient's performance status, and we take the objective response rate as a secondary endpoint, and we confirm that every patient showed a complete response to the treatment. And there is cohort 3 is remaining. It will be receiving 10 mg of VS1 plus RT, but in phase 2 clinical trials, we only including 2 mg and 5 mg of VS101. This is our second IND approval from the US FDA was available data for breast cancers. These data show that VS101's efficacy in the triple-negative breast cancer mouse model. We compared VS101 to paclitaxel, RT, and we found that 3 combination is the most effective. However, we think it's interesting. 2 combination, RT plus VS101, is similar to the 3 combination. It means that we can get a same or better efficacy using less drug. So I think it is very beneficial for patients in the future. Now we are going to explore a new potential of VS101 for rare and incurable cancer glioblastoma. Yesterday, we submitted the IND for phase 2 for the glioblastoma in the United States. And in this study, we compared VS101 to RT and temozolomide. Temozolomide is a standard of care of GBM. However, it has problems of the toxicity issues because it has a poor BBB penetration, so high dose administration is required. VS101, its API has been used for psychotic disease over 20 years. It has advantages as a brain tumor treatment. So you can see here, 3 combination is most effective. However, we think this study is interesting because it implicates the various applications of VS101 is possible as a radiosensitizer and also chemosensitizer. Our goal is to develop radiosensitizer for all lives with various solid tumors regardless of its irradiation type. To achieve our goal, we plan a comprehensive strategy to develop it. First step is repurposing an existing drug because it's very fast. Second step is developing a complex for the patients who require more potent treatment. Last step is develop a new molecular entity of radiosensitizer based on our research and know-how. And since the last year, we decided to research over the combination with the radiopharmaceutical therapy. So now we are actively seeking for partnership with the institutions and companies. Our target indications: head and neck cancer, breast cancer, and glioblastoma. Last one is glioblastoma. We expecting the IND approval available in the first quarter of 2025. We have a plan to, after the HNC in the United States, we just kick off the GBM study. There are the market is emerging. There's no approved drugs as a radiosensitizer, but few candidates are under development. The first movers are J&J and Merck. They licensed in the Jevinapan and MBTXR3 in the stage of 2. Compared to the other competitors, we are confident in 3 points. First, VS101 has multiple mechanisms of actions, including the MOAs for other competitors has. A second thing is, as a drug proposing, we are very safe. There's a low level of toxicity is anticipated. Last thing is, we are oral agent. It is very easy to use compared to the injection type radiosensitizer. We expect that market potential will be over $80 billion when radiosensitizer used for various solid tumors in the world. I'm very proud of my company because it continues achieving the milestones. We have demonstrated our capability of drug development by obtaining 2 IND approvals from the US FDA. Now, kick off our research collaboration over the radiopharmaceutical therapies with the Korea institution, which can handle the radioisotopes for the critical studies. And also, we initiated collaboration with the Bajaj University, which is the most famous institution over the RPT. Lastly, I'm very proud of that we having a great mentor, J&J, by joining the J-Labs company. These achievements are a result of the collective intelligence. It is created by mingling the internal and external great people. Our leadership team has a great experience in the Korean pharmaceutical industry, especially our CTO have experience of 4 drugs approval for the cell therapies. And also, our technology comes from the very famous institution in Korea for the radiological medicine, named Kirans. Our goal is by accelerating the drug development of VS101 in 2025, and as the result, we seeking for the licensing out of VS101. So the first priority is over the getting the interim data of phase 2 for head and neck cancer in 2025. So we are expecting we are expecting IND approval for GBM in the first quarter is available. To support our drug development, we are raising $20 million. After that, we can expecting after 2026, we can expanding our indications in the United States. Our funding status is here. $20 million will be used, such as $5 million to be used for operating the phase 2 operating the phase 2 clinical trial for head and neck cancer. $9 million will be used the R&D programs for other pipelines, and $6 million will be operating costs and spendings for licensing out activities. We have been raised money from the Korean capital market. This one will be our first overseas fundraising. If you are interested in the Korean biotech company, which will lead the next paradigm shift, let's connect. Thank you for listening.